chapter from my master's thesis when I was at University of Hawaii at Manila. It's been a minute since I've been to school, but this has been a really awesome opportunity to finally present this section. I haven't been able to yet, so let's get started. So this, like I said, was a chapter in my thesis, and Donald Drake, that's my advisor. Okay, so Hawaiian Islands, and we can extend this to island biology, have a lot of human impacts on islands, yet at the same time we have a very poor understanding of basic ecology of the native plants. You can extend this to a lot of native animals, and native organisms to islands in general. We can also extend this to basic understanding of ecology for native species on islands, such as plant-animal interactions, like pollination. This is very similar for the coastal habitat in Hawaii. Again, there's very few studies. There's just some basic studies that describe the physical environment and some of the vegetation, along with taxonomic descriptions of plants and organisms. And again, the habitat has been severely impacted by development, recreation, non-native species, habitat fragmentation. You can kind of keep going. And unfortunately, the human impact on island systems is ever increasing. So, for this study, we looked at plant Jacquemontia sandwicensis in the plant family Convolvulaceae, morning glory. We picked this plant because we were interested in coastal native plant pollination. Again, we didn't really know much about it. This species, Jacquemontia, came up as a pretty common plant and important plant in a vegetation survey that we were working on, along with a community description of pollination for coastal native plants. And this plant, like I said, was common, so we thought we need to do a more in-depth study on it. So what we started with, basically, is that it's just a common plant in terms of vegetation and the community level, a basic taxonomic description, and again, it's important plant in this pollination study that we did at Kaina Point. And you can see it's really important for albatross to stand on. But we don't even know that because, again, we're missing the basic biological studies on it. So I'd like to introduce you a little bit to what the coastal pollinator community looks like too. Again, it's poorly understood. We do know there's a native set of bees called hylias. They're called the yellow-faced bees. As you can see, they're just the cutest bee ever. They're the top right picture there. The last time I looked it up, it's 63 species of endemic hylias bees to Hawaii. They're across all the islands, from the coast to the mountains and the forest. They're throughout all of the Hawaiian communities, yet we don't know much about their basic ecology. Since they're a bee and they eat pollen, we would assume that they must be important for pollination for native plants. So back to this other study, this Kaina Point pollination network I've mentioned. We learned that Jacquemontia sandwicensis had a lot of visitors. It's a common plant, and one of its visitors was a hylias species. So this also helped gear this next study for looking at Jacquemontia and trying to find the native yellow-faced bees, the hylias, and see if we could learn more about native plants and native pollinators and their relationship. So Jacquemontia sandwicensis in this network study, it's this one up here, and you can see it has a lot of flower visitors. For other potential flower visitors, pollinators for native plants in this study, the rest of them are non-natives. couldn't find any other native pollinators in that study. So again, this comes from the impact from humans. There's some other graduate students, and there's a few publications that have looked at some other coastal native plants in Hawaii. For example, Sesbania tomentosa. That was studied by Hopper, and he also found the Hyleas yellow-faced bees. So we wanted to learn more about the importance of Jacquemontia in terms of its role in pollination, in the pollinator community. We saw that it had a lot of flower visits in the other study and a lot of floral visitors. And so we also wanted to learn how much does this plant rely on pollinators. So we decided to look at the breeding system for Jacquemontia and see, again, how much does it rely on pollinators and if they're even necessary. And then we also went out to the field to look at if there are pollinators. So we did a set of breeding experiments and we did some more pollinator observations. So our study sites, this is the island of Oahu in Hawaii. An interesting Arcana Point study is this corner. So we thought we'd go to the other side of the island and see what it looks like on the other side. And this area, you could just call it, it's a little bit more degraded. Arcana Point is a park and it has protection. So we thought it'd be interesting to see in a more degraded community what it looks like. So we have these sites that we went to. Okay, so we collected individuals for the breeding experiment from all 4 of these sites. We went to the field to look at pollinator observations for Makapu'u Cliff, Ka'iwi Shoreline, and Halona Blowhole. We didn't go to this beach park. There really wasn't any particular reason why, but that's what we did. So our breeding experiments, we did a field breeding experiment. So this was at Makapu'u Cliff. We did a bunch of different crosses. We also did a breeding experiment in the lab. And again, we did all these different crosses. We looked at fruit set, seed set, and germinated seeds. So this is some pictures, just basically what it looked like. We used the organza fabric just to cover the flower, so we could manipulate the pollen donor, or if we wanted to put pollen on it at all, so we could test for those. We simply would use a jeweler's tag as the fruit developed, just to keep track of it. And so this picture on the left, that's an example from the lab, and then this picture on the bottom, that's out in the field. And then these are the treatments. They were basically similar in the lab and in the field. I'll try to go through these quickly. So control is just your flower. There's no bag, there's no treatment, there's no hand pollination. It's just open to the environment. We had a treatment testing for autogamy. Just call it auto. So it was bagged. There was no hand pollination. There was no manipulation there. And we just wanted to see if it could self-pollinate. We did this. We did 2 versions of selfing. We had facilitated selfing. It was bagged, and the pollen from the flower was from the same flower. This was testing for self-incompatibility. This was done in the field because it was simpler, where in the lab we did this next version itself, where it was bagged, and the pollen came from a different flower but of the same individual. And so we could, we had a little bit more control in the lab setting, that we could get more flowers, and we could it was easier to control moving the pollen from one flower, that individual, to the next. We had an in treatment. So this was trying to see if there was some kind of inbreeding depression, and so it was within the population, and then out, which was out of the pollen came from an individual from a different population to see what our maximum potential seed set and outcrossing could be. Similar, so we did pollinator observations at the 3 sites I mentioned, just to get an idea of the floral visits. Okay, so we'll just jump into our results for the lab reading experiment. So I really enjoy how this came out. I'm going to show you the seeds per flower. I'll let you know that we also tested for fruit set, seeds, and the germinated seeds. They were consistent. So we'll go with seeds per flower for today. The control and the auto treatments, they were significantly different from the self, in, and out. And so in the lab, the control and auto, we were thinking this would happen because there aren't many pollinators up on the balcony where we did this. And the auto, there was no pollen getting moved onto the stigma. So we had the higher scheme treatments where pollen was getting moved onto the stigma. In the field breeding experiment, we had no significant difference. And this was fine too. We were expecting this. The control is now open to pollinators in the field. The auto treatment, it makes sense for the field site because it's so windy. And this flower has 2 long anthers that you can really imagine that as it gets beat around in the wind, that these 2 anthers are a similar length to the style and stigma. So you can imagine some seeds that actually happening that way. And then we had a selfing and an in-treatment within that population where the pollen was getting put onto the stigma, and we had seeds. And then we had the controls at 3 sites, and these are 3 sites also with the pollinators. And we didn't see much of a difference in seed set. Okay, and then just real quick, our pollinator observations. We had some diversity. Here's our hyleas, our yellow-faced bees. We have female, and then the males, which are hyleas and sinus. The females are distinct here because they're all black, and they're very difficult to distinguish in the field as which species of hyleas. So we decided the best way to distinguish it was Hyleas anthracynus and versus Hyleas female. And unfortunately, the rest of these are all non-natives. So Jacquemontia had some Hyleas visit the Makapu'u Cliff and also at the Kaiwi shoreline, not the Halona blowhole. And then overall, again, you can see there just weren't too many visits from the native pollinator. So what we've learned from this is Jacquemontia has this mixed mating system. It can outcross, but it looks like it can also self-pollinate in those odd instances where there's the right amount of wind and the pollen is, I think, falling from those tall anthers onto the stigma. So their reproductive success, it depended more on the pollen getting placed on the stigma rather than from which pollen donor. And then in terms of conservation view, the seed set is possible without pollinators, so it's a slight sigh of relief for that wind mechanism that I talked about. But the success is definitely higher with pollinators. And our pollinator community, it really is largely relying on those non-native pollinators now. We do have the one native pollinator. Hyleas is a story for another day, but they're endangered mostly. So they're relying on the non-native pollinators. So with that, I want to say thank you in Hawaii, mahalo. I'm happy to take questions. These are all my assistants and helpers. And I also want to say, I moved to Texas now, so I'd love to make some Texas Botany and Conservation friends, so feel free to come.